Hello, welcome back to your online video lectures. This chapter, chapter 15, we'll be talking about the chromosomal basis of inheritance. So in the previous chapter, chapter 14 was all about Mendel and the gene idea. We talked about the monk Gregor Mendel, who did pea plant crosses and started to figure out this idea of these heritable traits, which we call genes. But remember, that was occurring at the end of the 1800s. We really didn't start studying DNA and linking genes to DNA until the beginning of the 1900s. And of course, it wasn't until 1950 that we even knew what the shape of DNA was. So this is all in the beginning of when we're figuring this out. In this chapter, we're going to go into the chromosomal basis of genes, right, of inheritance. We'll talk about sex-linked genes. We'll talk about recombination. We'll talk about some chromosomal issues, differences, aneuploidies, and that will be about it for this chapter. For this chapter's objectives, students will model the processes and patterns of inheritance in eukaryotic organisms, particularly describe and model the fundamentals of genes and alleles, describe inheritance using proper genetics terminology pertaining to organization and transmission, predict the process of inheritance from numerical and/or pedigree data, produce numerical predictions and/or pedigree patterns for the processes of inheritance, and describe and relate the contribution of environmental variation to phenotypic variation. So let's just take it back a chapter. Remember in the last chapter that we talked about Gregor Mendel, the monk who was conducting scientific experiments by crossing pea plants and looking at a couple of different traits. So he was looking at flower color, flower position, seed color, seed shape, pod color, and stem length. And he was noticing these different patterns that were emerging, and he saw them via ratios. So one common ratio was a 3-to-1 ratio, which we see about, and it's not perfect because it's never perfect, but it's about a 3-to-1 ratio that always pops up when he does this type of cross. We have our P generation, our true breeders. We had a purple flower and a white flower. This is the end of the 1800s, so he has no idea. He cannot genotype these. He doesn't really know what genes they're hiding. When he crossed these two true breeders, he always got a purple flower no matter what. So the F1 generation, those hybrids were always purple. But then when he crossed two F1 purple flowers, he would get 3 purple to 1 white, and that's that 3-to-1 ratio. So it was crosses like this, and he also did it where he would look at 2 traits. So this was here. This we're looking at color and texture. And there was a new ratio that came here, 9-to-3 to 3-to-1 phenotypic ratio. So it was these crosses that began to give Mendel this idea about these heritable factors, which we now have come to know as genes. So when he first saw these heritable factors, they were just this abstract concept because he didn't know what DNA was. We didn't have that type of technology back then. Today, though, we know that these heritable factors are called genes. And more importantly, we know where they're located. They're located on the chromosomes within the DNA. Well, that code is coded within the DNA. We even have different techniques that allow us to visualize the chromosomes under special types of fluorescent microscopes. In this particular picture here, we can see the location of a particular gene can be seen by tagging isolated chromosomes with a fluorescent dye. So these chromosomes were all tagged with a orange type of dye, and then they were able to be viewed most likely under a fluorescent microscope. So how did scientists make the connection between the idea of an heritable gene and the fact that that gene would be on a chromosome? Well, Couple chapters back, we learned about mitosis and meiosis. Cytologists, so cyto means cell, so cytologists would be scientists that study cells. Cytologists worked out the process of mitosis in 1875 and meiosis 15 years later in 1890 using improved techniques of microscopy. So the question is, how did scientists bridge this abstract concept of this hereditary unit that Mendel discovered to DNA, to the chromosome? Well, luckily around this time, at the turn of the 19th century, the end of the 1800s, cytologists were also working out the processes of mitosis in 1875 and meiosis in 1890 because there's a strong correlation between our technology and then the discovery that comes with it. So we had some improved techniques of microscopy, so cytologists were able to work out mitosis and meiosis. Well, biologists began to see some parallels between the two, between the behavior of how Mendel described the hereditary factors and these chromosomes. And you can see in this picture over here, this would be meiosis. So we have in the P generation 2 true breeding individuals. We have a yellow round with homozygous dominant, and then we have a true breeder here, but they're green and wrinkled with homozygous recessive for 2 traits, right? We're looking at color and texture. Each of these individuals, these cells, go through a process of meiosis, which isn't shown here. So don't be confused. It's just assumed here. We know that the only genes that they have to give, that this one has to give, it could give a big Y and a big R. So this guy's gametes will only be a big Y and a big R. And this one over here, these gametes can only be a little Y and a little R. So we have a little Y and a little R. So here is a haploid gamete, and this is another haploid gamete. Each one is N, so this is N and this is N. We need to create a 2N cell. So these 2 gametes are fertilized and we create a new individual. Now that 2N individual, at some point in their life, will go through their own meiosis stage. So that's what we're looking at here. So we have our 2 stages of meiosis. Meiosis 1 and then meiosis 2. They're not really showing everything here. And meiosis is a very important process because we're creating these gamete cells, these haploid cells, which also have, the process has crossing over as part of it. So there's 2 important parts of meiosis, the law of segregation and the law of independent assortment, that the cytologists were working out. With the law of segregation, the 2 alleles for each gene, so of the red and the blue, have to separate in anaphase 1. And then for the law of independent assortment, this is talking about in metaphase 1, where the tetrads have to line up along the metaphase plate, and it doesn't matter if this is our genetic material. Independent assortment says that it could be where the reds are all on the left and the blues are on the right, or for one of them, the blue can be on the left and the blue can be on the right in the other. This will create 2 different outcomes and different gametes. And you can see when they're on the same side here, we have these as our outcomes, as our gametes, and when they just start with that one difference there, look at the gametes, we have a little bit of a shakeup in the genetic material. And this isn't even accounting for crossing over, which is going to create even more genetic variation. If you're confused about this, go back and look at the chapter, the meiosis chapter, which is chapter 13, because it's good to understand that process before coming over here. But this process gives a ratio that we've already seen with Mendel when he was crossing his pea plants, the yellow round with the green wrinkled. He was getting the 9:3:3 3:1 ratio for the dihybrid cross. So this is the same ratio, right? So this is where the cytologists, the biologists, were seeing the parallels between Mendel's work and the cytologists' work who were figuring out meiosis. So it was around 1902 when these particular scientists, Sutton and Boveri, were noting these parallels, and they began to develop the chromosomal theory of inheritance, which it just states that genes, that those heritable factors that we're talking about, those are contained on the chromosome. But of course, this is just a theory. It was just circumstantial evidence. So it wasn't until a little bit later, again, we're still in the beginning, the early 1900s, where we started to get some solid evidence. So we have Thomas Morgan. Morgan showed that Mendelian inheritance from Mendel has its physical basis in the behavior of chromosomes. So what did he do? First, we have to mention the model organism that Morgan used for his work. So he used a fruit fly, which has the name Drosophila melanogaster. So, Drosophila melanogaster. Fruit flies are a good, a convenient organism for genetic studies because they produce a lot of offspring. They breed every 2 weeks, and they only have 4 pairs of chromosomes. So this is great because if we're going to be looking at the genes, honestly, to figure out the very basics of them, the less that there are there, the better. So with these flies, Morgan noted that there is a wild type, which is a normal phenotype. A wild type fly is one that has red eyes. But after a little bit of breeding, Morgan got a mutant fly with white eyes. So when I say wild type fly, we're saying red eye, and mutant fly, we're saying they have a white color eye. Now, this experimental design that Morgan used for his experiment is really important because he's going to be crossing a mutant fly with a wild type fly, but he has a particular sex for each fly. So he crosses a mutant male. So the male has the white eyes with a wild type red-eyed female. So what he found, again, was that the F1 generation had all red eyes, and we're going to look at a graph of this in 1 second. The F1 generation all had the red eyes. So this is hearkening back to Mendel's experiments, right? When he had his true breeders and he crossed the purple and the white, and he always got purple for the F1. So Morgan always had red eyes for the F1 generation. That shows us that those would be the dominant trait. However, when he crossed the the F1 generation with themselves to create the F2 generation, again, just like Mendel we got a 3:1 ratio of red to white. However, only the males had white eyes. So let us look at this. Here is his crossing scheme. So his P generation, we have a wild type red-eyed female with a mutant white-eyed male. All of the F1 generation, whether they were male or female, all were wild type. So they all had red eyes. Then he would take a wild type female, cross it with a wild type male, and this is what he got. So we have the 3:1 ratio, 3 wild type to 1 mutant. However, he noticed that the mutant that he got was only ever a male. Now, this is important because this is showing that there's something strange going on. If you're not getting any white-eyed females, there's something within the mechanism that's special. So because there's a difference in what females will show versus what males will show, this is suggesting that this gene has something to do with their sex chromosomes X and Y. So Morgan reasoned that the white-eyed mutant allele must be located on the X chromosome. So this experiment, and we're going to walk through these slides showing the genes, but this experiment was able to correlate the gene to the chromosome. So Morgan's findings supported the chromosome theory of inheritance, which was first developed in 1902 by Sutton and Boveri. So let's walk through this cross together so you can see how this happens. Because again, this is different from what Mendel was showing with his crosses, with his monohybrid crosses with the pea plants. So we start off with two individuals, a wild type female and a mutant male. Because of the way that the males always were having the white eyes, this told Morgan that this is sex-related, sex-linked, and ultimately what it is that the gene, the allele for eye color is located on the X chromosome. So here's this wild type female who has two X chromosomes, right? Females have two X chromosomes and males have an X and a Y chromosome. The female is a true breeder, so she has two copies of the wild type allele, and the annotation for this is a capital W with a plus, so this is the wild type. So she's got two wild types. The male has one X, one Y. On the X chromosome, he's carrying the mutant allele, so this is just a capital W. So when these individuals create their gametes, the male is going to create the sperm, so each type of gamete gets one of these chromosomes. So one of them is getting the mutant allele, and that's the X chromosome, and here's the Y chromosome, which doesn't have any allele for eye color because it's only on the X chromosome. And then the female only can create one type. She has two copies of the same exact thing. So we don't have to duplicate. We don't have to have that twice in here because the proportion, the ratio is still going to be the same, even if you were to draw a second box for a second X chromosome. So because they're the same, we only need the one. So we have the X chromosome with the wild type allele. We're going to cross those in our Punnett square. We have a nice truncated Punnett square here. So what we create is a female with a wild type allele and a mutant allele, but because the wild type is dominant, this female is showing the wild type phenotype of red eyes, right? And the male here, so this individual got the X chromosome with the wild type and the Y chromosome with nothing on it. So again, this individual is going to show with the wild type phenotype. And this is why the F generations all have red eyes, regardless of whether they are female or male. This mutant allele is hiding in this female. So then when we cross this female with this male, again, we're going to separate the gametes. So this male creates a wild type red-eyed X chromosome and a Y chromosome with nothing on it. This female creates two different gametes. We have a wild type X chromosome and we have a mutant X chromosome. So here's our Punnett square for the F2 generation. We fill it in and we see that we created two females that are red-eyed, a male that is red-eyed, and here is where the mutant comes through, because this male received the Y chromosome from the father and that mutant X chromosome from the mother. This individual also received the mutant, but in addition to the wild type. So this one is showing red. So this is why the F2 results always showed two females, all of the females with red eyes, and half of the males will have red eyes and half will have the mutant white eye. As we move through this chapter, we're going to talk about linked genes, and you're going to hear sex-linked genes. And this means that that gene is located on a sex chromosome, so either the X or the Y. If I just said sex-linked, you don't know, but we usually will be more specific and we'll say X-linked or Y-linked. Now remember that the X and the Y chromosomes are homologs for each other. This being said, the X and the Y chromosomes are very different in their size and the kind of genes that they contain. X chromosomes are much larger than Y chromosomes, and Y chromosomes they only have a couple of hundred genes on them, and they tend to have genes that direct the development of male anatomical features. And the Y chromosome does have this region called the SRY gene on it, again, which is for directing of masculinization. So the homologous portions of X and Y would be the bottom part of X, because again, the Y is much shorter. So the bottom part of X and then Y, those are the homologous corresponding regions. So remember, again, females are XX, males are XY. An ovum, which is the gamete produced by females, can only contain X chromosomes, while sperm can contain either X or Y chromosomes. And a gene that's located on either of these sex chromosomes, again, this is sex-linked, and then we will say either X-linked or Y-linked to be more specific. So let's look at inheritance of the X-linked type genes specifically. So again, Y chromosomes mainly encode genes related to sex determination for males. X chromosomes, on the other hand, have some genes that are for characters that are not related to sex. For example, we have a couple down here: color blindness, hemophilia, Duchenne muscular dystrophy. And we'll see that X-linked genes tend to follow these inheritance patterns. We can have a dominant X-linked gene, or we can have a recessive X-linked gene. For recessive X-linked genes, they can only be expressed if a female has both copies, but for a male, they only need that one copy. So let's see, this is why, for example, males tend to have higher instances of color blindness, because the genes that result in color blindness are located on the X chromosome, and males only need one copy. It's a recessive gene. They only need that one copy. If a female has one recessive, one dominant, she won't be colorblind. So let's look at a couple of these Punnett squares so we can see that. We'll start with A. So in A, the female is homozygous dominant. So we are looking at X-linked recessive disorders. So let me just state that. Remember, dominant will have a capital letter and the recessive has the lowercase. And for the colors of the squares, white, no color here means that they don't have a gene that's for it, and neither do they have the trait. So the darker color is that they carry the allele and they express the trait. And the lighter colored here is that they carry an allele, but they don't express the trait. So this is a female that doesn't carry any of the, we'll just say color blindness, X-linked recessive color blindness alleles. This male has one of the alleles on their X chromosome and, of course, nothing on the Y. So the gametes are created. The female creates the dominant X, the X with the dominant allele. The male creates the recessive X-linked allele with the Y. When we create our Punnett square, we see that this combination of parents will create males where none of the males have a gene for it. They don't carry the allele, neither do they have the trait. However, all of the females here carry an allele but don't express the trait. So these females are called carriers. They're not expressing it, but they do carry, which means they would pass it on to their sons. Mostly. Well, we'll see that in a second. Notice how, if I were to just say, this is a question, be careful with how questions are asked on exams and quizzes, because if you looked at this, you'd say, okay, well, 50% of the progeny are carriers and 50% are not. That's true, but if the question is asking how many sons versus how many daughters, just be very careful of that. That question is geared toward sex-linked genes. So if we're talking about an X-linked gene, it would say what percentage of the sons carry the gene, and you might go, 50%, but no, it's 0%. So just be careful with how that's questioned. All right, let's look at B. So that would be, let's say, these two types of individuals. So we have a carrier and we have a non-carrier, non-expressing here, male. Males can't carry X-linked recessive. They either have it or they don't. But so we create our sperm. Here's an X with the dominant with the Y. Here's an X with the dominant, and then the X with the X-linked recessive. Here, the colorblind allele. So this combination makes a female that's not a carrier and non-expressing, a male that's non-expressing, and then we have a female who's a carrier, and we have a male who has the disorder. So this male will be colorblind. So again, if you were to see a question about this, you just have to be careful how you're going to answer it. If I say, what percentage of the females are carriers? You don't want to say 25%. You want to say 50%. 50% of those females. Okay, and then the final one would be a mother who's a carrier and a father who is colorblind, so has that colorblind allele. The sperm, we have the X-linked recessive allele here, the Y, and then we have an X that's dominant, and we have the colorblind one here. So this creates a higher instance of this occurring. So this female is a carrier, this male is non-expressing, and then this female is expressing, and this male is expressing. So again, just be careful how questions are worded with X-linked genes. And also, a lot of students mess up with setting up Punnett squares with sex-linked genes. Don't forget, if we're talking about the sex-linked gene, you have to put the X's, and then, you can create whatever letter you want for your superscript, unless it's given to you in your question. But I do have a lot of students that forget to do the X as their chromosome, because in the previous chapters, we don't put this on a chromosome. We just would do capital N, and, lowercase n, whatever it might be. So don't forget to use your chromosomes here, and these are superscripts on the chromosome. In mammalian females, there's a phenomena called X inactivation, because remember, these are females, so every single one of their cells contains 2 X chromosomes. So both of those chromosomes are going to be containing copies of genes, and we don't need duplicate copies of genes. So in the cells of mammalian females, one of those X chromosomes is going to be inactivated. So this is called, when that X chromosome becomes inactivated, what it does is it gets condensed. It gets closed up so that it can't be read, right? We can't get any type of cellular machinery into here because it's all closed up. So this is called a bar body. This inactivated X chromosome is called a bar body. The other one remains open and active to be read so that the genes can be created. So that the genes can be read and the proteins can be created. So this female cat here is shown with 2 different fur colors, both black and orange. And this is because fur color is located on the X chromosome. This particular female is showing both colors because she's heterozygous. So her cells, her 2 X chromosomes, one of them would be carrying a black color allele, and the other one will be carrying an orange color allele. And her X inactivation just across her body happened to different X chromosomes. So in part of her body, the orange was inactivated, and the other part, the black, was inactivated. And this gives her that tortoise shell look to her. So if you ever see a cat like this, now you know. You also will know it's a female because only females have the two X's so that she can carry two different colors. And just remember that these are called bar bodies once they are inactivated. Now, when we say the word linked, like if I were to say eye color and hair color are linked, what that means is that they are located on the same chromosome. They are physically linked. And the closer that two genes, two alleles are together on a chromosome, the more likely they are to be inherited together. Of course, we know that during crossing over in meiosis 1, we can kind of mix and match parts of homologous chromosomes. So this is where that linkedness comes together. Genes that are closer together, if they're mixed and matched, they're going to move with each other. And ones that are farther apart, they have more of a likely chance that while crossing over is occurring, that they'll be switched off onto another chromosome. So when we say linked, we mean physically, that they are on the same chromosome, and the closer that they physically are, the more likely they are to be inherited together, plain and simple. But we do have some math that we can use in order to figure out how close two genes are on a chromosome. And of course, what we could do is we could look at how often those two genes are inherited together. So let's very briefly look at this experiment. Again, this is by Thomas Hunt Morgan, except this time we're looking at the linkage of two factors for Drosophila that are not sex-linked. So this is nothing to do with the X or Y chromosome. This is just any of the other chromosomes. So let's see. We're looking at two factors. The color of the body and the type of wing. Color of the body and the type of wing. So we can see the first fly here is a homozygous dominant. So this is the wild type here, and this will be the mutant. And remember, in these crosses we always start with true breeders. So this fly here is homozygous dominant for both of these traits. Notice the annotation for these. The body is a lowercase b with a plus. So this is the wild type. The mutant body, which is black, a black body, is just lowercase b. And then for the wings, we have normal wings, which are VG plus. So this is the wild type. And this is the mutant, which is just VG without the plus. So the pluses, once again, are the wild type, and the non-pluses are the mutant. So when we cross this homozygous dominant individual with the homozygous recessive mutant, we get always in our F1 dihybrid test cross here, we always get an individual who is showing the dominant traits. So they have the dominant phenotype, the wild type phenotype. But if we look at their genotype, this individual here has one of the wild type, one of the mutant for the color, one of the wild type, one of the mutant for the wings. Okay. Then when we cross that individual with another individual who is the mutant, so this is a homozygous recessive, again for both black body color and vestigial wings. This is what we're going to be looking at for this test cross here. So just first look at the numbers. So we have two traits, body color and wing type, and they are linked, so they are on the same chromosome. So we have one chromosome where they're linked here and another type of chromosome where they're linked here. And we somehow get individuals later on that are mixes. So this individual is the wild type. They are gray with a normal type of wing. So this individual looks just like these individuals. And this individual here is the mutant. They are black. Sorry, this one here is the mutant. They are black with vestigial wings. So they're just like this individual. But we also have two new types of individuals that are a mix. So they have the normal body type with the mutant wing and the mutant body type with the normal wing. And if we look at the numbers, they have high numbers of individuals who look like the parent types, right? This is a parent and this is a parent. These two are just as the parents. And then we have these new types here, and these are called recombinants because we've taken those two genes and we have recombined them, mix and matched them, and they're at lower numbers. They're all around 200 out of over 2,000, right? So again, those individuals, those offspring that had the phenotype that matched one of the parents, that's called a parental type, and offspring that had non-parental phenotypes. So this is where we had recombinations, new combinations of traits. These were called recombinant types or recombinants. So these were the parental types, and these were the recombinants or recombinant types. We can look at the frequency of recombination of genes to tell us something about whether they're linked or not. If you have two genes on different chromosomes, so they're not linked, you'll have a 50% recombination. So we're talking about these numbers down here. These individuals had no recombination. These individuals had recombination. This is about 400 individuals, and this is about 2,000 or so, 1900. So that was much less than 50%, right? So this is not, these two genes are not on separate chromosomes. If we had a 50-50, where if there was 1,000 of these and 1,000 of these, that would let us know that these two traits were on different chromosomes. So we're going to be looking at the recombination frequencies because, again, they tell us how close genes are to each other on the same chromosome if they're less than 50%. When Thomas Hunt Morgan saw these combinations and noticed that there were some parental but some that were new combinations of genes, even though those genes were linked physically, he proposed that there must be some process that occasionally breaks that physical connection between genes that are on the same chromosome. And this is where the idea of that mechanism of crossing over of homologous chromosomes comes from. So now let's actually walk through that very same test that we saw that Thomas Hunt did. Let's actually look at the chromosome so you can see how this is occurring. So we begin with our wild type individual here. Two characters that we're looking at, body color and wing type. This individual is wild type for both, right? B plus, VG plus, B plus, VG plus. This individual is the mutant. So this is homozygous recessive for all traits, so just BVG, BVG, both of those homologous chromosomes. We cross these two individuals and we get this individual. These individuals will all have the same phenotype and genotype that's shown here, so they'll look like the dominant traits here. Even though this individual is heterozygous. So they have both the dominant and recessive for both alleles, B plus B, VG plus VG, okay? So then we're going to take a female from this group, from the F1 group, and we're going to cross this female with a male who is, once again, homozygous recessive for with all the mutant alleles. So we have BVG, BVG, okay? These individuals have to create their own sperm and gametes, right? Sperm and egg. So this individual is going to be going through meiosis, during meiosis, in meiosis 1, that's where crossing over occurs. However, these are the homologs, and this is where crossing over would occur. But if you switched the B for B, it's the same allele. If you switched VG for VG, it's the same allele. And we can never switch for different alleles. That's bad. We can't switch eye color for hair color, right? We can only switch the homologs, the homologous alleles. So this individual will go through crossing over in meiosis, but he won't create anything different. The only gamete that he's going to create is this sperm here, which is BVG, which looks just like this. So that's his gamete. This female, on the other hand, she's going to be going through meiosis, but her homologs each contain different genes. So some of these homologs are going to stay the same. So crossing over might occur over here. It's not going to occur over here. So in that case, this will look pretty much the same. So that would be this egg is possible, right? This red one, and this light pink one is possible. Those are the parental phenotypes that we were seeing. But for some of those, what's possible is during meiosis 1, there could be some crossing over between these guys here. So we could switch over here, it would be, we can switch VG for VG plus, right? So that's what we see here. We've switched this red little piece here, VG plus, which was previously on this one, and this VG, which was previously on this one. Just by doing that one incidence of crossing over, we've created 2 new chromosomes here, 2 new gametes. We have B plus VG and BVG plus, right? Those are the recombinants. These are parental, these are recombinants. So then we take these gametes, we cross them with the sperm that we created to actually create individuals. And what do they look like? We have this individual, which is, they're going to be wild type because they have 2 wild type genes and 2 mutant genes. The wild type is dominant, so they're going to have a wild type phenotype. We have this individual, which is BVG, so that's all the mutant. So these are all the mutant phenotype right here. So this individual is going to look just like this male. This individual is going to look just like this female. There's our parentals. Here are our recombinants. Now this individual will be, we have 2 VGs, so this is the vestigial wing. So we have the mutant wing, but we have the wild type body color of gray, and this is the opposite. So we have the wild type, the mutant body color of black, but the wild type wing type, which is normal, right? So here's our recombinants. Now, when we look at the numbers of the recombinants, we can add the number of recombinants, which, let's go back, 206 plus 185. 206 plus 185 is 391. So we're going to put that in the numerator on top, and in the denominator on the bottom, we're going to add all of these together. This is our total number. So 965 plus 944 plus 206 plus 158 gives us the grand total of 2,300 total offspring. So to find the percentage of recombinants, we take the total number of recombinants over the total number of offspring times 100 to give us a percentage. So these individuals, these 2 genes have a 17% recombination frequency. Again, that's less than 50. 50 tells us that they're on 2 separate chromosomes. These are on the same chromosome, so they're going to be less than 50. And the smaller the percentage of recombination, the closer those 2 are together. So if these 2 genes were twice as close, I guess, or if we bring this closer here, we would have an even lower percentage of recombination frequency. The closer they are, the lower the recombination frequency. It's going to go all the way up to 50%, and then once it's at 50%, that tells you those are probably on different chromosomes. We have to keep this in context of life and the continuation of life. All of this is to create genetic variation. We have these recombinant chromosomes which are able to bring these different alleles into new combinations to create new gametes. And then those gametes are able to be randomly fertilized. So if we have a bunch of sperm and a bunch of eggs that are created and they're all different, which ones become fertilized also has its own sense of randomness. And this all just increases our genetic variation. It gives us an abundance of genetic variation. And this is always good for us. Make sure you go back a couple of chapters. Make sure you know the three things that affect genetic variation. Okay, so a little bit of math. I just walked you through how to calculate recombination frequency. So we can take that number, that recombination frequency that we calculated, and we can create a linkage map. A linkage map is a genetic map of a chromosome based on recombination frequencies. Distances between genes can be expressed as map units, with one map unit representing a 1% recombination frequency. So, in the example that we just walked through, looking at body color and wing type, we calculated that the recombination frequency was 17%. So we take that 17% and we turn that into a map unit. We turn that into 17 map units. And here they are. This would be 17 map units apart from each other. This is another gene that if we were to do the crosses, count the progeny, and calculate a recombination frequency between these two, we'd see that there was a 9% recombination frequency. And then if we did it between these two, we have another 9.5% recombination frequency. So this would tell us that this is somewhere in between these two. This is a linkage map, just taking those recombination frequencies and seeing how they relate to one another as far as distance and location across that across that chromosome. Genes that are very far apart, the farther we move away from each other, the more this percentage increases. And genes that are very far apart on the same chromosome will have a frequency near 50. Once we reach 50, this tells us that they are no longer on the same chromosome, they're on different chromosomes. And again, just so that you have a mental map of this, we just saw that crossing over is what creates those recombinations. So if there was going to be crossing over occurring on this chromosome, since these two are pretty close together, let's just say that the crossing over was this area right here. They're close together, so they're more likely to stay together and not have much of a recombination. That's why they have a lower recombination frequency. These are farther apart. So it's still low, but they're still farther apart. So their recombination frequency increased. There's even more of a likelihood that these two will be separated than these two. So again, as we go farther and farther away from each other, our percent recombination frequency will increase. So now we know our heritable factors, our genes, are located on our chromosomes. Well, there could be a couple of bad things that happen to our chromosomes which could lead to medical issues. So we can have alterations in the number of chromosomes, or we can have an alteration in the structure of our chromosomes. Let's look at abnormal numbers of chromosomes first. So this is a picture of meiosis, meiosis 1 and meiosis 2. How do you have a cell, a gamete, that's created with an abnormal number of chromosomes, meaning we have one or two too many or one or two too few? Well, if we look at meiosis 1, we have two tetrads here, right? Well, they were tetrads. They're going through anaphase, so they're being separated. Anaphase 1. It's really important that those two tetrads, that those homologs are separated during anaphase 1 properly. If they're not, when the spindles go to pull them apart to the opposite poles of the cell, we'll have one side pulling nothing and another side pulling both pairs of homologs. This is called non-disjunction. Pairs of homologous chromosomes do not separate normally during meiosis. So we can have non-disjunction in meiosis 1, in anaphase 1, or we can have it in meiosis 2 during anaphase 2. And depending on where this non-disjunction occurs, will affect the gamete that we create. So let's look at this side first. We had a non-disjunction occur in meiosis 1. So both of these pairs of homologs went this way. Now just look at these cells after the first round of cytokinesis. This cell has 3 pairs instead of 2 pairs, and this cell has 1 pair instead of 2 pairs. So these homologs had separated, and then now these are sister chromatids of each other, right? These are sister chromatids, these are sister chromatids. They were lined up along the metaphase plate, and these are going through anaphase 2 of meiosis 2. And these are separating properly, but we already have a wrong number. This has 1 extra pair, and this is lost a pair. So the resulting gametes that are formed, there's supposed to be 2 chromosomes in each one. We have the letter N here. Remember N, we saw N stands for haploid and 2N stood for diploid. For humans, our N is 23 and our 2N, our diploid, is 46, right? For this cell, the N should have been 2, and it's not. We have 3 here, so this is N plus 1. This also has N plus 1, and this is N minus 1 and N minus 1. So what we see is when there's a non-disjunction that occurs in meiosis 1, all of the gametes that result have the wrong number of chromosomes. Now, if any of these gametes go toward fertilization, even if the gamete that they're going to be fertilized with has the correct number of chromosomes, that first cell, that zygote, is going to have the wrong number of chromosomes. This is called aneuploidy. Aneuploidy results from the fertilization of gametes in which non-disjunction has occurred. So if this goes towards creating a human zygote, that human will have an aneuploidy. Let's look at this over here. So in this meiosis 1, these cells have gone through the correct, the homologs have separated, right? Then these cells go through cytokinesis. Those homologs are lined up along the metaphase plate, and now it's their sister chromatids that are separating. This side, they've separated properly. On this side, however, one of the sister chromatid pairs isn't separated, so they're both going to the left, while on the right, this part gets none of this genetic material. So looking at the gametes that result, this is an N plus 1, because we have 3. This is N minus 1. However, these are fine. These are correct. So if we compare non-disjunction occurring during meiosis 1 versus non-disjunction occurring during meiosis 2, meiosis 1 is much more detrimental because every single one of these gametes is going to result in an aneuploidy, whereas only 50% of these gametes are going to result in an aneuploidy. These are perfectly fine. These are going to be bad. I have a couple of terms here, monosomy and trisomy. So if a zygote has only 1 copy of a particular chromosome, that is monosomic. So this this individual is lacking this larger chromosome here, right? Because this one got 2 of them. This one's lacking it. When this gamete is used in fertilization with, let's just say, something that looks like this. This is normal. This cell will have 1 copy of this long chromosome. That is monosomy. That zygote is monosomic for this chromosome, this long chromosome. Now we can also have trisomy. Trisomic zygote has 3 copies of a particular chromosome. So that would be, let's say, using this gamete. This gamete has an extra copy of the long chromosome. If we use this gamete and we fertilize it with a regular gamete, this will have 1, 2, 3 copies of the long chromosome. And the correct number of the short chromosome, right? 1, 2. We always want 1 from mom, 1 from dad. But this is going to get 3, 2 from, let's just say, mom and 1 from dad. So that would be trisomy. What we see here is a karyotype of an individual who has Down syndrome. Down syndrome is an aneuploid condition that results from 3 copies of chromosome 21. So Down syndrome is also known as trisomy 21. If we look at this karyotype, we see there's 23 pairs of chromosomes. There are 22 pairs of autosomes and 1 pair of sex chromosomes. Each of the 23 pairs of chromosomes in this karyotype are homologs of each other. And you'll notice that at chromosome number 21, instead of having 2 homologs, there are 3. So this is the aneuploidy right here. And this is a trisomic. There are 3 chromosomes. So this is trisomy 21. We can also have aneuploidy of the sex chromosomes. Non-disjunction of sex chromosomes produces different aneuploid conditions. The one that's mentioned here is Kleinfelter Syndrome, which is a result of an extra chromosome in a male. Particularly, males will have 2 X's in addition to their Y, so they are XXY individuals. We also have Turner syndrome, which is a monosomy X. In other words, these individuals are X with nothing. So they're females that only have the 1 X chromosome. These females are sterile, and it's the only known viable monosomy in humans. So those were alterations in the number of chromosomes. This is the alteration of chromosome structure. So if we break a chromosome, there can be 4 different types of changes in the structure. We can have a deletion, a duplication, an inversion, or a translocation. In a deletion, there is going to be a removal of a chromosomal segment. So in the box A up here, we see that, let's just say these letters correspond to genes, A, B, C, D, E, F, G, H. This deletion, we see that there's a removal of the D segment, so this chromosome is completely missing that gene or that set of genes that's there. So that's your deletion. We can have a duplication in which a segment of the chromosome is repeated. So you see the BC segment was duplicated and both of those were sewn in to this chromosome. We have an inversion. So instead of ABCD, we have ADCB. So this section was inverted. And then we have a translocation. Anytime you see trans, it means across or movement. So we have 2 separate chromosomes here, and what happens is a piece of each one swap. They translocate. A couple of disorders that result from structural alteration of chromosomes. There's a syndrome called the cri du chat, which is the cry of the cat, which is a result from a specific deletion in chromosome 5. Go ahead and Google this. There are some videos on YouTube. So with this, a child born with this syndrome is severely intellectually disabled and they have a cat-like cry, and they may die in infancy or in early childhood. Also, certain cancers, the one that's listed here is chronic myelogenous leukemia, CML, which are caused by translocations of the chromosomes. That's this one that we were looking at here, where two pieces of the chromosomes will swap. Lastly, let's just take a look at a few inheritance patterns. Particularly, we're going to look at exceptions to that, the normal Mendelian inheritance. Mendelian, having to do with Gregor Mendel, when we learned about dominance and recessive. There's a couple of patterns that are exceptions to that basic rule that he first founded when he was doing his pea plant experiments. So one of those exceptions is going to involve genes located in the nucleus, and the other involves genes located outside of the nucleus. In both cases, the sex of the parent contributing the allele is a factor in the pattern of inheritance. The first one is genomic imprinting, and this one has to do with genes that are found in the nucleus. So for a few of mammal traits, the phenotype is dependent upon which parent passed along the alleles for the trait. The variation in phenotype is what we call genomic imprinting, and it involves the silencing of certain genes depending on which parent passes them on. Let's look at an example for that. Here we are looking at the alleles for a protein called IGF-2, which is insulin-like growth factor 2. So this is a growth factor. So this is going to have to do with the size of the mammal that we're talking about here, which is the mouse. So there are two versions of this allele. There's a normal IGF-2 allele, which is colored in dark purple, and there is a mutant IGF-2 allele, which is colored in light purple. So we see here that this mouse has a normal IGF-2 allele from the father and a normal IGF-2 allele from the mother. With this genomic imprinting, only the paternal chromosome, the one coming from the father, is going to be expressed no matter what is on it. So it doesn't matter what the mother is giving, only the paternal chromosome is being expressed. So this particular mouse is going to express the normal IGF-2 allele, so this is a normal-sized mouse. However, these mice down here, it's a little bit different. This mouse received, they're heterozygotes. So this mouse received a normal IGF-2 allele from the father, but a mutant IGF-2 allele from the mother. This is okay because the mother's allele is not expressed, only the father's is, so this is a normal mouse. Here we have another heterozygote, except this paternal chromosome has the mutant IGF-2 allele, and this maternal chromosome has the normal one. The normal one, again, the mother chromosome's not expressed, only the father mutant IGF-2 allele is expressed. So this mouse is a dwarf mouse because they have the mutant growth factor here. So again, this is genomic imprinting. This is a gene that is located in the nucleus on the normal chromosomes, and it has to do with which parent passes along the alleles, and the other one of the parents, their alleles always gets silenced. So this particular example, the mother's always got silenced. It's believed that only a small fraction of mammalian genes are under genomic imprinting, and that most of the imprinted genes are important for embryonic development. And if you recall from chapter 4, when we were learning the functional groups, we learned about the methyl group CH3. And we learned that methyl groups can be used for gene expression, for controlling whether a particular protein is made, whether a particular gene is expressed. And really all that it is the CH3 group is very small, and another one that we can put on is an acetyl group, which is much larger. When we put the CH3 group onto there, it kind of closes up the DNA. So it's telling the machinery in the DNA, which would read it to make the protein, it's saying, no, you can't get in here. You don't need to read this. So when we methylate a chromosome, we're kind of turning it off. The other exception to inheritance patterns has to do with the inheritance of our organelle genes. And if you recall from chapter 6, we saw that not only are there genes located in the nucleus, but also in the mitochondria in mammals or in animals, and in both the mitochondria and the chloroplast in plants. And this is what gives us evidence to the endosymbiotic theory, right, of how we went from prokaryotic cells to eukaryotic cells. So if this is sounding new to you, just go take a look at that again, the endosymbiotic theory. There are some differences with the DNA that are in the organelles, though. As you know, the DNA that's in our nucleus is, should I just bring up this picture? This is the condensed form, right? They're linear. They're just condensed here because they're going through a part of mitosis. But they're linear. The DNA that are located in our organelles, in the mitochondria and the chloroplast, are circular, small circular DNA molecules, and we call these plasmids, but the DNA is circular. It's very interesting how we inherit our mitochondria. Our mitochondria are solely inherited from our mothers, and this has to do with when the zygote is forming. Remember, that is the very first cell. That is when the sperm is fertilizing the egg. So just imagine we have an egg, which is a cell, and it's a cell, so it needs energy, so it needs to have a mitochondria. We also have as many mitochondria. And then we also have sperm, also a cell, definitely needs lots of energy because it has a lot of swimming it has to do. So sperm have a lot of mitochondria. But when the sperm penetrates the egg, it has a special mechanism of how it releases its genetic material. But the mitochondria don't get transferred from the sperm to the egg. That new zygote gets the male DNA from the sperm, has the female DNA within the egg, and then has the female mitochondria within the egg. So that's why we only inherit our extranuclear genes, anything outside the cell, maternally. Of course, the DNA that the mitochondria carry, and also same for the chloroplast, is it's solely for that organelle. So it's going to have the DNA that's encoding all of the proteins that are involved for the mitochondria, for the citric acid cycle, for oxidative phosphorylation, for the electron transport chain. All of those proteins are encoded within the mitochondria themselves. You can have defects in mitochondrial genes, which would prevent the cell from making enough ATP. This is not good. So this results in diseases that affect muscular and nervous systems, which of course require a lot of energy. For example, here, mitochondrial myopathy and Leber's hereditary optic neuropathy. So just two examples of mitochondrial gene defects. Now, this is where it gets interesting, because with our modern day technology and our knowledge about the cells, we can bypass something like this one particularly, this mitochondrial gene defects, by using a two-mother egg system. So in this case, you would find a donor, a healthy donor that doesn't have any mitochondrial defects, to donate an egg. You would remove that person's nuclear genetic material, and instead transfer the prospective mother, her DNA, into that donor egg. Now you have an egg with the prospective mother and mitochondria from the other woman, but it's healthy mitochondria. And then you could fertilize that egg with sperm from the prospective father and transplant that zygote into the womb of the prospective mother. Okay, so that is it for chapter 15. I think some of the bigger things to take away from this chapter are to make sure when you look at this, you understand it, and you could walk through this with someone. You could explain it. You can have a mental picture of it, be able to compare and contrast this to the typical Mendelian genetics. So this is a sex-linked trait here. Definitely make sure that you know how to make sex-linked Punnett squares like this, answer questions about them, fill them in. Be able to answer questions about linked genes, understand the difference between a parental phenotype and a recombinant type. Be able to calculate the recombination frequencies. Here, recombination frequency, it's always the total number of recombinants over the total number of all the offspring times 100. That's how you get a percentage. Understand how the recombination frequency, what that can tell us about the distance between two different genes. Remember, if two genes are on the same chromosome, like this, the recombination frequencies will always be less than 50. The closer they are together, the smaller the recombination frequency. As they get farther apart, that recombination frequency increases until we reach around 50. And then that kind of acts, if you have two chromosomes that are really far away from each other, they pretty much act as if they're not on the same chromosome, as far as how often they're inherited together is. If you have two genes that are on two separate chromosomes, that recombination frequency is at 50%. You either will inherit them together or you won't. Definitely know the alterations of chromosomes. We can change the number or we can change the structure. There's always questions about non-disjunctions, what they are and what their result would be. You know, whether you have a non-disjunction in meiosis 1 versus meiosis 2. You can have a question, we have 50% of the offspring have will have a trisomy, where did this occur? Or if you see something like this, you have the numbers. So gametes have n+1, n+1, n-1, n-1. Where did the non-disjunction occur? You might see something like that. Number 2 common aneuploidies are monosomic and trisomic. An example of trisomy is trisomy 21. The chromosome number 21, there are 3 copies here. And then, of course, we have our alterations of structure. This isn't too difficult, right? We have deletion, duplication, inversion, and translocation. Depending on what it is, they could range in how detrimental they are. We could have something as serious as the Cri-du-Chat. We can have cancers. And then definitely understand the exceptions to the Mendelian inheritance rules that we learned. So these are a little bit different. They're not just your basic dominance and recessive inheritances. With genomic imprinting, it has to do, these are nuclear DNA, it has to do with which parent you receive it from. One of the parents will always be silenced. In this example, always the female. The maternal chromosome is silenced. And then with our organelles, the mitochondria is always given by the mother. So that's it for chapter 15. And then in the next chapter, chapter 16, we will get into DNA replication.